Morning, Joe.
Good morning.
Okay. So I'll just introduce the morning and then you can take over, okay?
Okay, that sounds fine. I have until 9, is it?
Yeah.
Okay,
I
great.
mean, and that's
Sounds good.
flexible. If people are asking questions, feel free to
Yeah.
drift. We've been doing that a
Yeah.
lot,
We're getting a lot of
Okay.
questions, yeah.
Okay,
which is great.
good.
Yeah.
Ask away.
Can you hear any background noise on mine end? We have a bit of planes that fly over where we
No,
were.
nothing.
Okay, so all right, great. Good.
Yeah. It's a bit chilly outside this morning though.
Yes, it is a little chilly outside. Nice change of pace. Yeah.
Hmm. I mean, I like to go out to breakfast
Oh,
and
yes,
I
that
had to
is
put a
lovely.
coat on.
Yes. Oh, yes, that's true. It doesn't feel like almost August.
No, it's July and temperatures dip below 60.
Yeah. So how many folks did you have the first couple of days? I see
About 80.
80. Okay,
Mm.
that's very good.
Hmm.
Excellent.
I actually noticed the first day you had 100 for
Oh
A.
yeah, it went. Yeah. Actually, what it did go up. It was a bit higher on the first day. Yeah,
Oh, that's good.
yeah.
So what part of your lecture did it start to dip?
The beginning.
Or maybe I should say, maybe it wasn't you. Maybe it was Lucian.
What
Yeah.
was it that like drove
Yes,
30
right.
people away? Like, oh,
Exactly.
enough of that, man.
Well, I certainly do that many times. I'll come on to something and say, yeah, I don't know. It's a little more than I can take. I think I'll go back to coffee.
Yeah. So Kimberly is on the. Kimberly's one of our our enthusiastic questioners.
Excellent.
Says it's a tough course, but we're still here.
Good for you. Okay.
Okay. Let's see. Give it another minute, I guess. Yeah. I mean, if it really is a toughish, that suggests there's room for a more introductory level
Hmm.
class.
Well, yeah, and I think it's been sort of mixed. I think it might be helpful to target it, like to say like this is for
Yeah,
people who know this much or something. I mean, it's nice to be able to say like everyone come and we'll figure out what the level of the people that come are. But when there's 100 people, it's going to be mixed. So
yeah,
And sometimes it's hard to be specific so people can
yeah,
self-evaluate. It's this thing of, you don't know what you don't know. Like,
yeah,
how do you rate your background in Python?
right.
oh yeah, Central Time. I'm sure some people are actually here, actually in the morning, are actually in the early hours of the morning.
Yeah.
Wouldn't
Oh,
surprise
yes,
me.
that's true too. Right. That's always, I mean, but it's a
And
challenge
if they
from
know
time to
the
time.
recordings are going to be available, it's like I don't have to be there live. I'll just watch it when they
Oh
go
yeah.
up.
Yeah, right.
So
Okay. Okay, I guess we'll start. So good morning or afternoon, evening, everybody.
mhm.
This is the last day of lectures. Tomorrow to be the hackathon, and then Friday it's invited talks. Um, Today, let me share screen briefly. This is roughly the what we're going to do. And as you've realized, we sort of run out of time or whatever, but that's fine. We've been very active. The whole viewing group has been active in asking questions and so on, which is fine. So first thing, we'll have something on model calibration. So I'm going to do something on signaling pathways, but I need to finish off the oscillators, and then I'll have something about signaling pathways. So I may go over a little bit. I mean, we've got full 4 hours if need be, and this roughly adds up to 3.5 hours. So we have a half-hour spare. I'll definitely break 11:15 on my time. So so that we have a break, and then John has his time. John Gennari has his time. Okay, so that's what we're going to do today. All right. In that case, I'll hand it over to Joe.
All right. Well, good morning, everyone. Or again, whatever time zone you're in. For us, it's certainly morning. Let me see if I can get myself set up. Get to the slides. Okay, so here is the folder for day 3 model calibration. That's where we are. And I'm going to start with these slides here. Okay, so far, I think that I don't know how much has been discussed about model calibration. Has that been referenced?
Nothing.
Or parameter calibration, they're equivalent ideas.
Yeah, nothing other than
people have talked about it. People ask questions, and we'd be like, "Yes, that's an interesting question," and we haven't covered it in the slightest. So,
and I showed them a crazy question. We talked a little bit about ChatGPT, and the other day I asked it to write a script to fit a model, and it did, including generating synthetic data, etc., etc. So,
cool.
but that's as far as we got. Okay, so they don't learn anything else.
All right, great. So at least we've got the basics, which is what I was sort of hoping for. And I'm sure that there are folks who've done this already and have an established workflow, and others who this may be their first encounter other than a conversation with ChatGPT for doing this. So so my first slide is a little bit just to sort of set a perspective here. This whole problem of model calibration, which is about figuring out values for parameters which you don't have, really good measurements for, under physiological conditions, it's challenging. The computational requirements for this can be tremendous. I lived in Arizona for several years, and it's subject to flash floods, and very common that you come to a road and it looks like it's flat, and you see something that's water there, and you don't know how deep it is. And that's very equivalent here, is that you just don't know how much compute time something may take, or some other issues you may encounter. So for those of you who are already familiar with some of the mechanics of what's involved with model calibration, it's really about estimating parameter values. I'm taking a step backwards and looking at the whole process. So hopefully you get something new out of this as well. And for those of you who've never encountered it before, hopefully this gives you a perspective about what's involved here. So let's start with a specific example. So this has been my sort of poster child and reference point, this particular model. It's one from biomodels. It's a model that was developed by Jenna Wolf, primarily in, over 20 years ago, almost 25 years ago now, about oscillations in glycolysis with yeast. And you can read some part of the abstract over here. This study required, and as most studies really do in systems biology, the studies, these are mechanistic models. You've been developing mechanistic models for something, for example, like mass action. You typically have, you're going to have the product of the concentrations of the reactants times a constant. The question is, where does that constant come from? Michaelis-Menten, you have a couple of constants there. You find that very quickly, you're going to have maybe 2 or 3 constants per reaction in your model. And typically you're using different constants in different reactions. So you pile up a lot of these constants. And the question is, I mean, that some of these, there are some constants that have been estimated under in vitro conditions, but to get physiologically meaningful constants is a real challenge, and typically you won't have that. So the trick is to use empirical data that you've taken, from observation, and run your model evaluating different values of these constants to see which combination of values then produce good values for estimates of the experimental data. That's basically the premise that underlies this. And what I'm going to go through is sort of some of the workflow involved with this and some of the challenges in practice with doing it. So here's the Jenna Wolf model of glycolytic oscillations. It's a model-sized model, although it's complicated enough so there's some very interesting behavior. And you see here what a total of 11 reactions, but it includes really the external feed of glucose. So really, depending on how you want to count it, maybe it's only 10. And if you look at then the rate laws involved here, you see a variety of different rate laws. Some of these are reversible. You'll see some things that look much more. This one flows over the other side. This one's a complicated one, J4. Others are going to look, for example, down here, J5. It's the way many people will name their constants. They'll name them by reaction. So this is J5_K time glycerate 3-phosphate times ADP. So this would be a classic, if you look at what the reactants are. So this would be mass action. And you see similar, variations on different reactions here. If you go across here and sort of add up what all the constants are, so here are all the constants. So we got, I think it ends up being around 16 constants, including that input flux. Maybe it's 17 with that. And if you choose the right value of the constants, you get these behaviors. So presumably she was trying to demonstrate that in certain cell populations, you can get these oscillations. So the question is then, with all these constants and all these places, how do we come up with reasonable values for them in a way that doesn't take a long time and it's meaningful? Okay, so in essence, what we're doing is we're doing this empirically based on combination of empirical and then some computational evaluation, trying to find the best parameters to fit the data. And for those of you who maybe have some statistics background or have encountered this term, the key concept here is something called the residual. And in statistics in general, there's a lot of negative thinking that rather than making the model look good, you try to make whatever you don't fit in the model, not so bad. So it's a strange idea. So with the residuals are what's left over after you take out what you fit. So in our case, what we're fitting is our mechanistic model, our tellurium model, an SBML model. So that's our fit with the values of the parameters. And what we're going to do is for each one of the floating species that we're estimating its time course, and we subtract that from the time course of the observed data, and what's left over is the residuals. And we're trying to make the residuals as small as possible. Smaller the residuals, the better the model. That's the philosophy here. Okay, so here's sort of a generic, algorithm for how do you do this fitting. So the input to doing this, you got to have a model. You've been looking at SBML models for the last several days. You've got to have a bunch of parameters that you're going to estimate. So in the Jenna Wolf model, we saw there were 16, 17 parameters, and you need observed values. So with these three inputs, then what you're going to do, you're going to produce some estimates of the parameter value. So these are the names of the, here we've got the parameters we're going to estimate. And what we're going to come out with are values associated with one. So this might be J1K5 is the name of one of the parameters to estimate. And over here, we might say, okay, a good value would be, 33. I'm just picking a number. So that's what we're going to do there with the output. So here's what we need to do. So we've got this, and again, this is, if you're not familiar with the term, what we call pseudocode. So this is all in English, but it sort of looks like code, and you could probably step by step translate this into something that was actually in a programming language like Python or C or Julia or your favorite language. So we're going to keep on going until we get good parameter estimates, or we've got some sort of a runtime budget. I don't know what it is. It may be in terms of dollars. It may be in terms of time. So a setting is an assignment of values to the parameters. So we've got our 16, 17 parameters. We're going to set values to them. So that's, of course, a question like, how do we come up with these settings? So that's going to figure that out in a minute. Then we run a simulation. Now we can't do step 1B until we've done 1A, because you can't run the simulation without having values for the parameters. So we're sort of making guesses up here. We run our simulation, then we look at the difference between this is the observed data that we came in with. We subtract off what we estimate from the simulation results with this assignment of parameter values. And then here we're just doing an evaluation to see how good it is. So a typical approach to looking at goodness in modeling is to look at the sum of squared error. It's, sort of like roughly kind of variance estimate. You want to square it because that way negative, if you're below the estimate, that doesn't cancel out something that's above. Sometimes people use the sum of absolute value. And so that's another one that has somewhat different characteristics in terms of the fitting. But something like that, typically we will use sum of squared errors. And then you have to have some criteria about what does it mean to be small. This is a really tricky one, surprisingly so, with biological models, because our units vary so much. They can be in fractions of a millionth, or it could be in hundreds of thousands. So it's a little bit tricky here to try and figure out what this one is. But this is the basic flow here, and this is what you're doing. You can see there are a number of places where there's some thought and potentially innovation that's required for your particular problem. The part that I'm going to focus on here is going to be this one right here. Picking the settings, the new assignment of values for the parameters to estimate. And this is a kind of search algorithm. We'll also see this called an optimization algorithm. It's trying to find, it sort of incorporates some of the later steps too. So the best, but how do you figure out that next value? And there are a couple of different techniques. Well, there are more than a couple. There are many different techniques you can use there. Okay, so you got it. So we've set the stage for what it is that the problem is that we're trying to solve. And I was just sort of scanning here to see if anybody's actually on the video feed. I think everybody I see is on video mute. Okay. It's I'm used to teaching to a live classroom. So every now and then, some facial, there we go. We get somebody here. Thank you. Thank you, Lucian. I appreciate the performance. Okay. So I'm going to thank you also. Anybody who can go off video mute, I appreciate it. So here's a couple of concerns. The first one is typically these models, as I mentioned, have a large number of parameters. 2 or 3 per reaction is not uncommon. And, there are many models, mechanistic models, that will have, 30, 40, 50 reactions. I mean, it's not a very large number because we sort of don't know how to deal with many of these, but they can have a large number. So we've got a lot of parameters, and typically the biological data sets that we're getting, we get a lot of numbers back. Like you may get numbers associated with every gene in a genome, but you don't get many samples. You may get, 20 samples, 40 samples, especially for a time course. So we actually don't have a lot of data for the number of parameters we want to fit. There's a classic sort of meme that comes from John van Neumann, that, with 4 parameters, you can fit an elephant, and 5, you can make its trunk wiggle. I mean, sort of that the thought is that, there's always a concern with having many more parameters than we have data that can fit. Just as a sidebar, I mean, this is one of the really big concerns about many AI models. You'll see like, ChatGPT and Llama, they'll have billions of parameters. So, where's all the data come from to estimate those parameters? All right, so that's concern number one. We're just sort of inherently, we're fitting a large number of parameters with not a lot of data. Okay. A second concern is something called computational complexity. This is sort of a computer science fancy way of saying it takes a long time to do what we're doing. So I'm going to give you a sense of this because I think this goes back to that picture I showed at the front about the cars submerged in water because it wasn't clear how deep it is. This shows you that even for modest problems, if you really want to try and resolve, estimate what the parameters are in a systematic way, it is a non-trivial exercise. So let's suppose very optimistically that Lucien has optimized Tellurium to the point where it's 1 microsecond for each simulation run. So each simulation run, that means that we've assigned values to all the parameters. That's a simulation run. In order to run it, you got to have values assigned to the parameters. Okay, so here is a scale that we're going to look at. We're going to look at we're going to do parameter fitting, parameter calibration. And rather than looking at the actual time in regular units like in hours, I'm going to look at the logarithm of the hours. So that means that if it's if something takes an hour, the value is 0. If it's less than an hour, obviously it's going to be negative. If it's more than the hour, this is why, because we're going to deal with very large numbers. So I'm going up here on my scale up to the age of the universe. So the log in hours of the logarithm of the age of the universe in hours is 14. So just to give you the scale we're talking about here. So let's take a look at what we might be doing here with parameter fitting. So over here, we have the number of settings. This is the number of different values we're assigning to the parameters. So if I have, one parameter with, and I'm considering 2 values, in log units, this takes me, minus 9.3, so log hours. So, much smaller than a second. Obviously, as I move up the number of parameters, then it's going to take longer, and you see that by the color coding. So, if I only have two different settings for the parameters, I'm still, in the sub-hour range, around in seconds. But the fact is that we're going to consider a large number of settings. Now, this is what's called an exhaustive search, where in advance, we pick what the different values are and we pick them in combination. I mean, the search algorithms don't work this way. They will, navigate and figure out some next value. But, that's almost always a heuristic. And so if you want to do it, systematically, you have to do it exhaustively, and just shows you the challenges involved here. So with the Jenner-Wolf algorithm, we're around at 16 parameters, and, say you want to at least explore, I don't know, 8 values, 16 values, we're in this, century range, multi-century range already, if you want to do this exhaustively, with this optimistic thought that we could, run a simulation in a microsecond. So this is extremely computationally intensive, and so you have to be able to figure out systematically what you're looking for. Okay,
Bill, you
so
have a question?
Oh, I'm sorry. Oh,
No, it's
okay. Go ahead.
From
What's the
Ayush,
question?
looks like. I don't, he has his hand raised.
Oh,
on
a
the
hand
Zoom
up.
thing,
Yeah,
so
okay, fine. Okay, Ayush.
Unless it was an accident, but Ayush, if you have a question, go ahead and ask.
You're on mute if you're not aware of that. Okay, I guess we'll assume it was an accident. Feel free to come back and ask again later if I overlooked you. Okay, so so how do we look at this problem, and how can we make it so that we can get by the seemingly intractable problem of a just really daunting computational requirements? The one way that people know about that works pretty well is to look at something called the fitting surface. So what this is if you consider, and best illustrated by example, actually. So let's consider a very simple model where we have, some floating species, that we this is that we're going to look at. We're going to look at this value of X over here that we're trying to estimate its floating concentration. And we've got two parameters, K1 and K2. We've got some, it goes to the X0 and X1 are fixed. So this is just basically where it all goes over here. And this one over here is a source, and that's the sink. So that's what's going on here. What we're focusing on here are the values, not so much the floating species values. We're looking at K1 and K2, and what are good values for K1 and K2? And so let's take a look and see what that looks like. So for this particular example, I generated some synthetic data, and the synthetic data assumed that K1 and K2 are both 1. And so what I'm plotting over here is if I look at different values of K1 and K2. So this is a controlled experiment because I know what K1 and K2 are. But suppose I move away from K1 and K2. I want to look at this, what I call the fitting surface, which is what does the root mean square of residuals look like? So we talked about the residuals before. Now we're talking about this measure, this squared variation measure. So that's the RMSE. And the darker it is, then the larger it is. And that's bad because we want residuals to be small. And the closer we get to that red dot, the smaller they get. Now, again, this is a simple computational example. So, it's going to be pretty easy to follow. But the thing I want you to focus on here is really the insight into search for this particular, for this particular model. And that is, if you look at this very closely, you see how it's dark here and it's getting lighter and lighter, dark here, getting lighter and lighter. And you can see a band of almost white. If you take a sort of different look at it over here, a different scale, you can see it more pronounced. Here's the red dot, and things sort of move in. All of the darkness turns to lightness right close to the red dot. This is extremely nice kind of problem to solve using something called gradient descent. And if you've done any AI stuff or any statistics, very familiar with this. And what that means is it's like climbing a hill, although I guess maybe descending a hill might be a better analogy here. And that is, you just take the path that gets you down the fastest slope. So you want to go down, as much as here's dark, you want to find the path that gets you lighter, 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 and then that's the bottom of the hill. And that's for a fitting surface. So the surface here is described by these color variations. Dark is high, light is low. That's for a fitting surface that is what's called convex. So convex is like conceptually in 3 dimensions, you'd think of it like a bowl that, you start anywhere on that bowl and you just follow it where it goes down fastest. You'll eventually get to the bottom of the bowl. And what is special about a convex surface is that the local minima, and so in this case, it's like here I'm right here and I'm going down and that's a local minima, or it seems local right there because I'm going down. If it's a local minima, that means it's the smallest point, the lowest value. The local minima is also the global minima. And that is a very important property that allows you to do this gradient descent and actually find the minimum value. And there's a whole theory around this idea of convex surfaces and what's called convex optimization, about how you do this and what functions are convex and what functions you can apply. So if you have a convex surface, and we have an example here of a model with a convex surface, you're in great shape because all this complexity stuff, it just vanishes. You very quickly find the local minimum. Of course, the big question now is, well, what's the reality behind biological models? Are they convex? So let's take a look at one. We're going to look at the Jenner-Wolfe model, the glycolytic oscillations we were talking about before. Take a look at that. So like I said, it's sort of like a roadmap to find and so by going down the slope here, look at these values, we're getting values for K1 and K2. Okay, so let's look at another model here. So here's the fitting surface for the Wolfe model. Now, there are 16 parameters here. So, my visualization is 60. Oh, I see there's a question. Let me pause for just a second. Before evaluation. Okay, so it's a question about what units you should. I mean, this is a great question. That the units are really challenging, and there are trade-offs either way. So one question might be, I'll go back here to show this for a second. So there's a question. I mean, like I designed this model so I could work in the same units. It may be that maybe K1 works in units of 10 to the minus 6, and K2 works in units of 10 to the plus 6. So it would be really challenging. There are a lot of different ways you can deal with that. You can sort of normalize it. You can use different scales. I find that this tends to be problem specific. You have to see something that makes sense there. It also presupposes that you sort of know the unit you're going to work with. We'll come back to this a little bit later, because this is one of the subtle challenges with doing parameter fitting, is that your search algorithm has to know the domain, the values over which to search for the parameters. And if you have something, if you don't know that, and you've got to work in a range of 10 to the minus 6 to 10 to the plus 6, that's a huge problem, because at 10 to the minus 6, you want to take very small steps, but that's going to kill you with 10 to the plus 6. And 10 to the plus 6, you want to take big steps, but it makes it unusable at 10 to the minus 6. So those are challenges.
I can also say that assignment rules are one of the things that people use to transform the model to an observable. Like you have things that are happening in the model, and then you use an assignment rule to transform that into something in the actual units and whatever else of the thing you're actually observing. Herbert mentioned like transforming concentration of hydrogen ions to a pH, or like I did the sum of everything into something. So maybe you can measure like all of the ATP in the system. So you measure like ATP in this plus ATP of this as an assignment rule. So you can either do that by exporting all your data and then transforming it, or you can just do it in the model itself with an assignment rule.
Right. Yeah, that's a good point. That's another important aspect of doing this in practice. I see. Okay, yeah, so there's a comment here by Anastasia, hopefully I pronounced her name correctly, about the value of log units. That log units are great for when you have a wide range of things that you're looking at. The one challenge with log units with biological models is some of these some of the values we deal with do have naturally 0. And so logs don't work well for 0. Okay. So let's take a look at the fitting surface for the wolf model. So what I've done here is, again, I'm just considering 2 parameters because I can't look at 16 space. So these were 2 parameters for the model. This is for the second reaction. There's a K over here, and this is the first reaction, the Ki. And what I did here is I just did a whole bunch of simulations. I set this to the value in the original model. So that's the red dot, like before, the quote true value from the model. And then I just considered all sorts of values around that and did simulations for them, and then looked at the root mean squared errors that I came up with, and this is the map. And so remember before, we had this nice property where around the red dot, it was very light. And as we got further away from the red dot, it got progressively darker. And that allowed us to do gradient descent. So we could just follow our way from dark to light, and we would end up at the red dot. This is very different. You see all these ridges over here where it sort of looks like it's light there, and then it gets dark, and then it gets light again and dark. These are what are called valleys. I mean, literally, that's the technical term. These are called valleys, where if you use gradient descent, you'd get stuck right there, because this is the local minimum. And you remember that it's only with convex surfaces that we have this great property that the local minimum is equal to the global minimum. Here's our global minimum. but we get stuck in this valley over here. You can see other places where it's sort of light for some reason over here. And then, other places, well, over here, we could probably make our way out of this region somewhat, but then we'd end up, maybe somewhere stuck over there. There are a few different techniques, and now we get into some of the various clever adaptations that people will use. So one thing that people will do if they still want to use gradient descent is they'll do what they call random restarts. So where you start on this surface makes a big difference. If I'm lucky enough to start right there, I'll find the red dot. But if I'm unlucky enough to start right there, I'm way away. So if you do randomly a whole bunch of starting points, then maybe one of them will be close enough to the red dot that you get it there. The problem is there's a classic term in statistics called the curse of dimensionality. And that is, as you add more and more dimensions, that your ability to get close to something in space drops radically. And so we have 16 dimensions in this case, because that has 16 parameters. And so you potentially need a very large number of random starting points before you can find that parameter. So yeah, it's a strategy, but it has limitations depending on the size of your model. So this is what I'm saying, gradient descent can get stuck. So there are a few things here in terms of what it is that's going to affect our ability to do this in practice. And it's sort of a summary slide here. So the nature of the fitting surface. So if I have a nice fitting surface like this, which is convex, I can do it very easily. This surface here, I'm not going to be able to use gradient descent. And I have to use other techniques as well, like random restarts. There are other heuristics that seem to work pretty well. There's an algorithm called differential evolution, which adapts the parameter values as you progress and tries to navigate through the surface. I mean, there's no real proof that this works well for any particular surface, but the practical experience is that it can work well. It's also computationally more expensive, so there's a trade-off there. So that's about that. So the number of parameters, obviously, if I'm dealing with fewer parameters, then it's easier to do. I have a smaller space I have to search. And this is one of the many arguments why simple models are better, because you don't have to fit as many parameters. Another possibility, although this I'm going to generalize a little bit, is the number of different values of the parameters that you're going to consider. Now, really, we're not doing exhaustive search practically. I mean, we're going to do with some optimization techniques that goes through things. But what practically this ends up meaning is, can I define the range over which I'm going to search? I gave before that example of small units versus large units. And so the ranges that you want to search across, that makes a big difference. There are iterative techniques where you might start out with something which is a fairly big range and sort of see where things settle and then try and narrow down your range progressively. And that's another strategy can be used. Where you start in the search, or if you start in many places, that's another thing. I mean, typically the argument that the resolution of this one typically is, I need to have a lot of random restarts, and maybe you'll get some idea about regions in which you can get better estimates, and maybe you sort of focus on a region. So that interacts with this idea of the bounds. Obviously, noisier observational data is going to be more problematic. Which species you're looking at, that can help you or hinder you. And the last one here, which I haven't really covered in this discussion, but really needs to be mentioned in passing. And that is that sometimes you'll encounter a situation in which you really don't have the data or the model to resolve a parameter value. You can imagine if, for example, in this Jenner-Wolf model, if I don't collect data on certain species, and I don't have data that sort of depends, that infers those species, then how can I possibly estimate the value of the parameters? Typically, the way this shows up is as you do different runs, you'll get the same fit for out of it in terms of the root mean square error, but with very different parameter values, maybe vary by a factor of 1,000. And that's because really that parameter is not influencing the part of the model that you have data for. And so that's typically the way you find out. And so identifiability means you just really don't have the data or the nature of the model is such that you're not going to be able to fit that parameter. And that's sometimes occurs. Okay, I see a bunch of questions here. I don't know. Lucian, can you? Okay, maybe someone can give me a summary. Is there something I should answer here? I see a bunch of questions.
Yeah, there is one that you can answer.
Okay.
Somebody asking, what's all the white area on your 2D plot?
Okay, so the white, okay, let me go back to the 2D plot because these plots are, I think if they are understandable, they provide particular value. Let me go back here. So, okay, I gotta go back. Let me go back to this one right here. So this is really a 3-dimensional plot here. And the 3 dimensions, so we've got the K1 and K2 value. The 3rd dimension is the root mean square error, and that's encoded as a color, really a grayscale. So a very small value will be white, a very large value will be black. And so all the white area right here means that all these values here are pretty small, but the smallest value is right at the red dot. So did I answer the question you were asking? Let me know.
No. What is the white area?
White outside the black.
Yeah,
there.
you have all this white area.
Oh, this area right over here. Okay, what I did was I just all I did was I rescaled the same plot. You see the values over here, this goes from like 0.6 and this is 0.6 to 0.14. So I rescaled it so you could see it's basically zooming out, but just on this part, just on this right here. So this section here, if you zoom out, you see this. That's
I think the answer is the white would be black, except you're not showing it.
That's correct. And that's probably a better way. Thank you. Yeah, that may have been a better way to show this. Yeah, I probably just should have shown that all black rather than or cut this out somehow. Maybe why did that? Go ahead.
Is that the same for the other plot too? I think it's the other plot as well.
Yeah, I think the other
next
plot
one,
as well.
yeah.
Yep,
all that white
it's the same
down
thing. Yeah, the white here is not that this is all goodness. Yeah, I probably just should have made that all black. I just didn't explore that space. That's all. It's an unknown. So okay. I didn't boldly go. Yeah.
I feel like you actually did explore the space. It just wasn't included in this color bar on the right. It was outside that range.
Yeah, that could be. That may be also that I limited the color bar. Okay, did that clear up that confusion? Hopefully. Okay.
Yeah,
probably.
Yeah, I think so.
Okay. Yeah. Let me know if that's still a point of confusion. I hadn't thought about that. Okay. All right. So let's see here. I see one. Okay. Maybe the better questions. Oh, is the absolute minimum the best? That's an interesting question. So what we're doing, if I understand the question, or at least my interpretation of it, is that this process here I described is finding the minimum value of the root mean square error. And the question is that really the best answer to this finding values of the of the parameters? That's an interesting question. There are a number of things that you might also want to consider. So one of them is what is your uncertainty quantification? It may be that okay, it's the minimum value, but there's a lot of uncertainty around that, and so that might be one possibility. There are also questions about how much you trust the model itself. Maybe there are variations on the model you want to consider, but with the same parameter values. So I guess the answer is not always.
I would say also, like in real life, data has error from a variety of sources that you don't know what it is. And the true value of the parameter basically has a likelihood of creating your data. If you get into Bayesian analysis, you talk about the likelihood of the data given a parameter space, given a set of parameters. And any one set of parameters might have the highest likelihood, but that's not means that it is true. It's just is the single most highest likelihood.
Right, right. So there's another one here about well, you have the smallest value for the search technique that you've used. That doesn't mean it's the minimum. It just means that's the smallest value you've encountered, and that's the practical fact.
But even if you did
right.
find the actual minimum, that you found the actual global minimum, that doesn't mean that that's the true value of the parameter. That just means that's the
absolutely
minimum.
Right. It doesn't. Right. Exactly. It doesn't. Right. That's all it really means. And this is there's a premise here that, you know, if I've minimized my distance from my model to my, you know, my experimental data, somehow it found the true value. Well, there's no reason why that has to be true. I mean, we're going back to the beginning where we talked about we have a large number of parameters, not much data. There could be many combinations of parameter values that give you that same minimum. So you don't know. So, but that's the premise. Yeah, you could. The problem is, yes, you could do something like this transformation. It potentially changes some of the relationships. But that's I would suggest, you know, trying a number of different things to see. Again, I think it's somewhat idiosyncratic. Okay. All right. I think we covered the questions, unless there's. Let me pause for a second. Oh, okay, almost covered the questions.
Yeah, someone mentions that like outlier points can skew your data, like especially if you're using root mean squared,
Right.
then that puts even more weight on the outliers. So you can just root mean
Absolutely.
absolute.
Exactly. Right.
Less,
right.
or
And
you can
these
just
are
discard your outliers, or like there's all sorts of
right. There are
techniques
a
there.
variety of things that people do in
Yeah.
these cases. That, yeah, this is why it's, you know, a combination of art and science. So yeah, these are good points and all part of the larger process. Okay. All right. So with this in mind, I think what I'll do is switch over to these slides that I have. So let's see, I've got it's a little crowded right here. So if we go back to the folder, I'm going to switch over to these the parameters fitting exercises and solutions. What I'm going to do here is give you a taste at least. Got about 15 minutes. Give you a taste at least about how you do this. You know, actually do this kind of fitting. So that is this Jupyter notebook here. And I assume everybody's been working with Jupyter notebooks already. I don't know, and I may not be current on the right way to do this now, because I know it's changed a little bit over time. It used in my version of this notebook, you actually have to restart the session. Is that still the case?
Yep.
Yeah.
You pip install everything, and then you restart, and then you can go.
And then you restart. Okay, fine. We're still there, and that's this NumPy thing, or I
No,
guess
it's
it
not.
was another.
Oh yeah, because Colab comes with a certain version of NumPy, and that version is upgraded. So if you don't restart, the front end of NumPy is the old thing, and the back end is the new thing.
Okay, gotcha. Okay, fine. We're in good shape there. Okay, let's see here. Okay, fine. So yeah, I think there's some detailed comments there that Herbert's addressing. Okay. All right, so let's do the run. Start and run all. Okay, get that going. Okay, so let me tell you how the notebook is organized. I'm not sure how much have in the notebooks you've been using, do you take advantage of the table of contents?
No.
Okay, let me explain
what
I do. I mean, I do a lot with teaching from notebooks, and so I find it very useful to take advantage of some of these text features that are available in notebooks. So, for example, this particular cell right here, it's a text cell, and you'll see it has a pound sign here. It uses Markdown. For those of you who are familiar with Markdown, basically about all you need to know is you can create a top level heading with a pound sign. If you want to create a second level heading, you make it 2 pound signs. And with that, you've probably have figured out, you know, 90% of what you need to know for this. So I create sections here so that it's easy to move around. So I'm sorry, did someone say something? Okay.
No, I don't think so.
All right, so easy to move around. So I typically organize my notebooks where there's some preliminaries, and typically these are going to be things like, all of my imports. I may have some helper functions, stuff like that. The notebooks are that you can hide. It's a little awkward with this arrow here. I find that in a bigger notebook, it's actually much easier to use the table of contents to sort of move around. So you'll see this. There's some preliminaries here with installs, with imports, some helper functions. This down here is worthy of looking at a little bit. So to demonstrate parameter calibration, I'm going to use a very simple model, a sequential pathway, or sometimes it's also called a linear pathway. You basically have a series of floating species, one that produces the next. And that's the sort of the running example. I also do the Jenna Wolf example. If you do it using the I mentioned in passing this search algorithm called differential evolution. If you use that algorithm, you get some better results for the most part, but it takes a long time. And so I've commented out that particular invocation with the differential evolution later down, just so that the notebook will run. I mean, right now we're still running. This little dot over here tells you that the notebook is still running. Okay, so this is our working example. And what we're going to do, if you remember the basics for fitting, what we need is we need a model. So we've got a model right here. Oh, the other thing I'll mention in passing, because this could be a little bit confusing. So we see the model over here with the rate laws, K1, and they have the constants. It's all mass action, K1 through K4. I set these all to 0 just because they're nominal values, because we're going to do fitting. We want to try and find out what the right values are for some separately obtained data. Have you discussed this idea of producing synthetic data? Has that already been discussed?
At least in passing.
Okay, fine. All right, fine.
Like we've done it. I don't know if we really explained it to the.
What bit?
Okay,
Sorry,
fine.
what bit again?
Producing synthetic data.
Yeah.
Oh yeah, no, we haven't. Yeah, no, we haven't really said anything about that.
So separately, I've created some data where what I did is I had actual value, non-zero values assigned to K1 through K4. I just picked values. I don't know. It's probably like something creative like 1, 2, 3, 4. So something like that. I created, so that would give me a model, and I can actually produce value, floating species output. And then what I did is I took that data and added some random noise to it. And it's that, synthetically produced data that I'm going to use as my observational data. It's not really observational data. It was created synthetically, but it gives me something to evaluate the parameter fitting algorithm. Okay, so what we're going to do is I'm going to go through at a high level some of the first few steps, and because there's a certain amount of detail here, and I want you to get to what I think is the important part, which is how do you navigate through the large number of choices you have about running these, running parameter fitting, running the parameter calibration, given the computational complexities for real models. So, so some of the things you can calculate the residuals, which is you pick, in theory, you have some assignment of values to the parameters, and then you look at the difference between what that produces for values, the time course of floating species, versus your observational data, which in our case is synthetic. And then you can look at different values of K1, see how that varies, just to get a feeling for what the model is. And that's really all that's going on over here. It's showing you calculation of the residuals. And then there's a certain amount of this where we get into some more details in terms of Python programming, in particular with arrays and the way Python does what's called slicing. And again, I don't have time to go through the details here. I don't know how much detail you went through, but for those of you who are interested, that's what's going on here. So I'm going to go down through here to the Okay, this is actually calculating the residuals. And then you can see here's what it looks like. These are the residual values. So what we've got now is we ran the model for certain values of K1 through K4. And then we looked at the differences between those values and the observational data. And actually the values that are assigned here for this run to compute the residuals were the true value. So we've got the true value, and then we've got the synthetic data that was produced from those same true values, but added some randomness to it. And what you see is you can get a sense that even if you hit it exactly right on the nose for this very simple example, you'll still see that the residuals, ideally you want 0 residuals, right? So that's where you want to go. So you see 1 through 4, mostly 0 there. And then with 5, you get a lot more variation. And I'm not sure that 5 was actually, I think, yeah, okay. So, and that has to do with the nature of this particular model, but this is not uncommon, especially at the early stages, that you'll see a lot more variability. So this shows you even under ideal conditions, it's not like you're going to get an exact 0. All right, let's go on down here, and I'm going to go here. So the second part of this is what we call the optimization. This is where we're choosing a way of fitting the parameter. So this is sort of like our choice for the next value of the parameter. So typically what we're doing is we're taking advantage of some kind of an optimization function. So let's start sort of from the end, and then move back to see the details involved here. So differential evolution is this fairly sophisticated adaptation. It's called a non-linear global optimization. It's global in the sense that doesn't look, it tries to do the whole fitting at once. And so you've got some function that you want to minimize. So in our case, the function is a simulation. And then you've got bounds on the parameter values, and it has to know, obviously, the parameter values themselves. And then it calculates the residuals out of this, and you want to try and figure out values that are minimum. And so you sort of work your way backwards and figure out, okay, so what do I need to do to prepare for this? Well, I've got to have the function to minimize. So in our particular case, the function to minimize a simulation, which means then that we have to then set that up to in the simulation itself. Okay, let's take a look at an actual model to give a feeling for. I've just got a few more minutes. I want to get you a feeling for what it is that actually goes on here. So here's that wolf model again, and so I've just printed it out, the antimony. And now we have the parameters. This is all of the 16 or so parameters. There's a certain format that's going to use for this. The package I'm using is something called SBSTOT, something we developed in the Saro lab. For those of you who do this, many people are using PyPesto. So PyPesto would be an equivalent to SBSTOT, and PyPesto takes advantage. Okay, I see a raised hand somewhere. Okay, why don't you go ahead and talk with the raised hand?
Raised hand's gone again.
Okay, all right. Okay, I see some things in chat. Is there anything I should answer?
Just that there are different search algorithms.
Okay.
The gradient descent is one, like genetic search algorithms and stuff.
Okay, that's
I was
fine.
wondering about our opinion about them. My opinion is, like, if they work, that's great. Like some of them work well in a variety of situations. Some of them work well in highly specific situations. You have to sort of know your problem domain and figure out like what kind of search space you are searching.
I mean, I can give 2 recommendations
Yeah.
from our own experience and our own work. And we've fitted some fairly large models, is differential evolution and swarm, the swarm algorithms. Those are pretty good, mainly because they are really good at searching the landscape.
And the only other variation I have on that is that, at least for the models we play with, because we teach this too, the only other thing I would throw in there is I think that gradient descent in combination with random restarts, and you may need a lot of random restarts, can in some circumstances be very effective. Yeah. Okay, so this step right here, where we're specifying the parameters and their values, this is a key step in terms of a way that you're in your workflow that you're going to do this iteratively. So we see if we don't have advanced knowledge of these various parameters, and we set these values here. So, what's the lower value? What's the upper value? So some of these parameters, you can see the wide range. Now let's see, we've got 10 to the 5th over here. That's almost 10 to the 6th. And then down here, I guess I see a 1. So we've got basically from 1 to 10 to the 6th. So I set the upper value here as 10 to the 5th. So for each parameter, you specify a lower value, an initial value, that's value here, and an upper value. So, 16 parameters, you got to do this 16 times. So what do you do? And so if over here, I'm telling you what the real value is. And sometimes, mostly, let's see, we're not going to catch it on this one because that's almost 10 to the 6th. Oh, wait a second. No, that's 10 to the 5th. I've got my decimal point wrong. Actually, we will catch it because that is 10 to the 5th. Okay. So, Bauer sort of knew it in advance, but this is something you're going to iterate off of. And you'll find that, something, if the result is a parameter estimate, it's at the highest range that you allow for your bound, the chances are you need to increase the bound. You probably haven't made it large enough. That's one hint that works pretty well. I find because you're going to do this iteratively, it helps a lot to sort of encapsulate everything into a function. I assume you had some at least passing acquaintance with this, with your, the introductory material. Typically, what you're looking at is you want to look over the time range you're considering, because that makes a big difference in terms of which parameters get fit better or worse. The fitting method, so differential evolution, swarm, least squares, the parameters you're going to, and the parameters that you're fitting, and this is this whole mess over here. So this includes lower bound, upper bound, and initial value. So all those are things you're going to vary during it. And then here are the kinds of outputs that you can expect from this. So this is really an exploratory data analysis. So you need the ability to plot the results. So here are the various floating species that come out of the fitting process. The dotted values are the quote true values. So again, these are synthetic data. We ran the model, added some noise. So this is what we come up with. And then the red line are the parameter estimates. That's what gets fitted. That's what we get when we fit. So we're trying to come up with values of the parameter estimates, but what we can plot and evaluate are actually the time course of the floating species. So there's a little bit of indirection there. So what we see here, for example, is a lot of variability in this oscillatory behavior in the, in quotes, observational data. And then the fitted data seems to get that early part, somewhat, but doesn't capture all the oscillations. So on these particular examples, some of this looks, this data here, this is going to be sort of hard to fit. Okay, so that shows you for this particular, I think in this particular one, it was using least squares. So now if we run it with differential evolution, I guess on this particular one, we don't see much difference. Okay, and then down here, we start to get better mean values as we vary it. So now what are we doing in our workflow? In our workflow, I think we chose, we chose some different variant. So now you see we're changing our upper and lower bounds. So we can get better values there. And then the story ultimately ends with running differential evolution, which sadly I had to cut out because it took too long. But then we actually do get good fits to all this. It actually, in this case, sort of is the hero or heroine that saves the day and actually gets us the fits. But the theme here throughout is that this is an exploratory analysis. You'll find that you will do so well on fitting some species, not so well on doing others. You have to decide which errors you can tolerate and even quantifying what well means, because there's some data points here that are sort of big and the curve doesn't go up there, but you know, how do I interpret that? Yeah. Okay. See, how am I doing? I'm a little bit over, not horrendously over, and I see 9 questions. What do I need to talk to? Or 9 chats. I guess
I
Herbert,
think
you've been
Herbert might have
answering.
been answering
Yeah,
things
I
as they
think
come
Okay.
up,
I'll
but yeah.
answer, yeah.
Okay. All right. Fine. All right. So I'm going to stop here. This is a topic with a lot into it. I really, one of the great resources you can look into is there's a lot of material on this package PyPesto. I encourage you to look at it because there's tutorial information around that. It's mostly about how you run and use PyPesto, not the sort of the Gestalt kind of ideas we're talking about here about what is model fitting and what are the limits of it. But for the next level of detail to do this in practice, I think that's a great place to go, and it seems to be a tool that's getting to be more widely used. So any
One of our
other
speakers on Friday is from PyPesto. So
excellent.
Yeah.
That'll be excellent follow-up. Questions on this point? Anything we went through today? Actually, you know, one of the things I would like is if somebody felt, I mean, this was only an hour talk and I chose to focus on sort of higher level concepts. I'd like to hear if, you know, maybe some of you were frustrated. Okay, yes, thank you. So Bart included a link to PyPesto. Excellent, thank you, Bart. Okay, all right. Okay, so I guess I will stop sharing at this point.
Yeah, okay, thanks. I'm just trying to find an email. Let me just see if I can find it. It's from a group I work with. Actually, just started working with, I guess, in this particular people, but I can't find the email. But they're fitting a very large model. And what they're using a swarm algorithm. And basically, a swarm is just multiple mini fitters that are trying to sample the landscape to try to find the minimum. And what they've done is because it would take a long time to run, because these things can take a long time to run, they use a specific Python package that distributes the swarm across a large number of compute nodes. And sorry, I'm not on video here. Across a large number of compute nodes. And this is how they get scaling. This is how they get to scale the problem. And I think it only took like a day, I think, then to fit, I think. It was a very large model. Not too large, really. But it was a very large model, like a couple of hundred parameters. And they use this swarm algorithm, and there's a specific Python package, I can try and find it, that they use that let them distribute the swarm over like a couple of hundred nodes, I think, on our UW cluster we have here. So, and that worked quite well. The other advantage of the swarm is, something we haven't talked about much, is, of course, you might get parameter fits, right? But then how certain are you of those fits? I mean, it could be really uncertain, right? So the idea is, can we get some uncertainty information? And it seems that the swarm, each individual in the swarm will have tried to do its own fit, and it'll be slightly different from every individual in the swarm. And so you get a sort of distribution for the parameters just from the swarm itself. And they're using that to infer confidence in the parameter values. So let's say there's a particular parameter where the swarm has every individual in the swarm says something quite different about the parameter. In other words, the confidence in the parameter value is then very low. Whereas another parameter, the swarm has a fairly high consensus on the value of a particular parameter, and therefore you might suggest that parameter was well estimated. So I'll try and dig that up this morning to get it to you. It's in an email, so I just need to find it.
someone guessed that it might be the number package that is the parallelization thing.
No, it's not number. Number is for jitting Python, I think. It's for making Python faster, right?
So just
Anyway.
comments just briefly on this. So so doing things in parallel, I mean, you definitely can get, and that's a great idea. And these packages have become more and more accessible, which is great. The one thing I want you to keep in mind, which hopefully came through in that chart showing, how long it would take to run parameter fitting. It's almost a drop in the bucket, even for modest models. If you go scale, I used to work at Google. We had several million cores. I mean, yeah, that's great to have all that compute available for you. But if you're talking about something that would run, basically a time duration of the age of the universe, okay, so maybe it's like only 0.99 of the age of the universe. It at its core, these are really computationally intense problems, and you need a strategy. You can't, and so the extent to which there is something like a gradient descent or something else that takes advantage of the nature of the landscape, the sampling idea of the landscape is good. You get a feeling about where you are. You have to. You're not going to defeat this by just getting more compute. It's far too big of a problem. You have to have a strategy.
Oh, yeah, so I got a little bit. He didn't actually give me the package he used, but they use 600 CPUs. They distributed the swarm across 600 CPUs, 600 cores, and I didn't say how long it took. Oh, 13 hours.
Cool.
It's 2,500 parameters. I don't know how.
Oh my God! Yeah,
I don't know how
I mean,
reliable that fit is.
yeah,
But,
I mean, but to put it in perspective, I mean, we're using these, AI models with many billions of parameters.
oh,
So
I know.
yeah,
It's
so I don't know
crazy.
how to interpret
Yeah.
that. So.
The other thing to point out is most of the time is taken up in solving the differential equations. Okay.
Right.
And one of the reasons why we actually developed Roadrunner, or it's one of the reasons I pushed for the development of Roadrunner, is to relieve that bottleneck. And Roadrunner is probably the fastest simulator you'll get, at least in the SPML world. And it can make a significant impact on how long it'll take you to
Absolutely.
do a
Right.
fit. I mean, other simulators are like 10 times slower than Roadrunner. In fact, that's why it was called Roadrunner, one reason, because it's Roadrunners are fast, right? And and if something is 10 times faster, and this one took 13 hours, it would take like 130 hours, which is what, 4 days instead. And if the model gets even worse, it could take weeks. So, and PyPesto, which we'll hear about on Friday, they are adding the option, or have added the option for you to use Roadrunner alongside their fitting algorithms in order to get the speed up. So.
And PyPesto also allows you to use a large number of different optimization algorithms, which is really your search. And as Lucian mentioned before,
yeah,
it's really idiosyncratic, these landscapes. We can't even visualize them because they're in so many dimensions. And so you want to have the opportunity to try different search algorithms.
Yeah, right.
Okay.
I think PyPesto's native native simulator is Amici, which also does like compilation down to machine code, but it's works on fewer platforms. So I think Roadrunner is easier
It's
to
awkward to
install.
set up.
So, yeah.
You have to have a compiler ready. It's technically challenging.
Yeah.
I mean, I know one of our postdocs, Michael, he had trouble getting it to work properly. It took him a while to get it to work. So, okay. Shall we have a quick break then? You can have a 15-minute break, grab a coffee or something. We'll go for a walk. Now that it's warmer outside. All right, so back at well, for me it's 9:30, so it's on the half hour, okay, for everybody else. Okay, see you soon.